All right, so we're going to finish off the digestive system with this lab. So we stopped at the stomach last time. So we're going to talk a little bit about histology of the digestive system, and then we're going to finish off with the small intestine, large intestine, rectum, and then also the accessory organs of the digestive system as well. So for histology, we're going to look at the esophagus and the small intestine. And if you think of the entire GI tract like one long tube, we're going to have some layers of that tube. And pretty much the layers are consistent throughout the GI tract. So no matter if you're in the esophagus or the small intestine, you're going to have the same basic layers. So you have the mucosa, which contains the epithelial tissue. And in the small intestine, you have these little villi. So they're like little fingers that protrude up to increase the surface area. So we'll take a look at that. And then the submucosa, so that's kind of where all the blood supply is, innervation, all that good stuff. Then of course we have a muscular layer as well, which contains the smooth muscle. So we have two layers of smooth muscle throughout the GI tract, and maybe a little bit more if you're in the stomach, you've got an extra layer of smooth muscle. And then you have an outside, an external layer of either serosa or adventitia, just depending on where you are in the body cavity. So that's your connective tissue layer on the outside. All right, so if we take a look histologically at the esophagus, we're going to look at the structure on the left, right? So it's a little different than the trachea. So the trachea is nicely held open with that cartilage ring, but the esophagus can kind of constrict a little bit. It's got that layer of smooth muscle. So if we blow that up on the right, you've got your internal layer, the mucosa, with that layer of epithelial tissue, that stratified squamous epithelium, non-keratinized. Then you have your layer of submucosa under that with some vessels and glands and things like that. Then we have our two layers of smooth muscle. So one's going longitudinally, and then one is going kind of right at you, right? So kind of horizontally. So it's coming at you or going lengthwise. So two layers of muscle, and then our outer layer of the adventitia, that connective tissue. Now, if we take a look at the small intestine, it's a little bit different. Specifically, you see all of these kind of long finger-like projections. Okay, well, these are the villi of the small intestine, so it increases that surface area for absorption of nutrients and everything. So you kind of can follow these villi, and the epithelial tissue is along those villi. So that's our columnar epithelial tissue. And then we've got that submucosa underneath and our layers of smooth muscle as well. Okay, and then so now that we're in the abdominal cavity, it's called the serosa on the outside. And if you notice, we were in the thoracic cavity with the esophagus, and that was the adventitia. So it just depends on, again, where you are in the body cavity. So we ended at the stomach last time, so we're going to continue with the small intestine. So the stomach empties into the small intestine, and that first region of the small intestine is going to be the duodenum, or you can say duodenum if that helps you remember how to spell it. That's the British version. And then the other two sections that are longer than the duodenum are the jejunum and the ileum. And then the ileum is what is going to connect up to the large intestine. And it's going to open up into the cecum, which is going to be the first part of the large intestine. So we have a valve. So again, we've been talking about kind of sphincters and valves all along the digestive system because you want everything to be going in one direction. So you don't want it to come back once it goes into the large intestine. We close that ileocecal valve, and then it can no longer back up into the small intestine. And if you look at the inside of the small intestine, we've already talked about villi as how the small intestine increases surface area, but there's actually these circular folds of the mucosa as well that help to increase the surface area as well. And we'll talk a little bit more about that in lecture. So let's look at our model. Okay, so we have the stomach up at the top, and then we have that pyloric sphincter that's going to, be an opening and a closing door into the small intestine. So that very first section, it's the shortest section of the small intestine, but arguably the most important for digestion, because that's also where we have the pancreas and the bile duct coming in, the pancreatic duct and bile duct to help the secretions digest the food. But then that duodenum turns into the jejunum. And then at some point and along the small intestine, it's not like you can say, here exactly is where it switches from jejunum to ileum. You can't really say that. I think the jejunum is longer than the ileum, but there's no designation that this is the exact spot where it changes. You can tell histologically, but really if you're looking at it grossly, you can't tell. But then that ileum is going to end at the cecum, and then we have this ileocecal valve. Okay, so here's the cecum here, and that's the first part of the large intestine. And that's where in other animals, especially like herbivores, they have a very pronounced cecum, and that's where a lot of their fermenting happens because they have a lot of microbes in there and they can digest that fiber way better than we can. But we do have a cecum, so we are able to digest, a little bit of fiber. We do have microbes in our large intestine that helps us to do that. So we do have a cecum. And then here's the appendix kind of shooting off the end of that cecum there. So if we take a look at another model, we kind of blow up the histological side of it. So if we look, this is the small intestine. So we're going to have these villi. Again, we kind of look at them like little fingers, and then there's going to be that simple columnar epithelium lining the villi. So that's all part of the mucosa. We have the submucosa where all the blood vessels and nerves are coming in, all the lymphatics. So remember we talked about that lacteal that comes in to absorb all the fats from our nutrients. So the fat goes into the lymphatic system in the small intestine, which is how we digest fat. So even though our sugar and protein goes into the portal circulation in the liver, the fat goes into the lymphatic system first. And then you can see these layers of smooth muscle in that muscularis externa. So these guys are kind of coming in at you, kind of a circular layer, and these are the longitudinal layer that are kind of lengthwise. And then of course it says the serosa is not in the picture, but that would be that outermost connective tissue layer. So even though we call the large intestine the large and the small intestine the small, the large intestine is actually shorter in length. The largeness is more referring to the diameter of the intestine. So once we bypass through the small intestine, we're going to dump into the cecum, which is the first part of that large intestine. And then the rest of the large intestine we split up into regions, just depending on kind of the curvature of the large intestine. It looks kind of like a big upside down horseshoe. So we have an ascending colon that's kind of coming up from the cecum, up kind of towards the liver, and it's going to make a turn, and we call that turn the hepatic flexure because it's turning right at the liver. And remember, everything having to do with the liver is hepatic. And then it's going to make a cross over the front of the abdomen, and we call that the transverse colon. And it's going to make another turn right at the spleen, so we call that the splenic flexure. and then it's going to go down. So we call that the descending colon. And then right at the end of the colon, where it turns into the rectum, it makes this little kind of S-shaped curve, and we call that the sigmoid colon. Okay, I think of like S as a S shape for the shape of the colon, and it's the sigmoid colon. And then that's what's going to turn into the rectum. So let's take a look at the model. So if we started down here at that ileocecal valve, okay, so here's our cecum. That's going to turn into the ascending colon. It's going to make that hepatic flexure turn. So if you think the liver is sitting kind of right up here and the stomach, right? So those are where those are all sitting up there. And then we're going to go transverse. I think of it like you're crossing a T, right, for transverse. And then you're going to make another turn at the spleen. So here's the spleen right here. And then you're going to go down the descending colon, and it's going to make this little S-shaped curve down here before it gets to the rectum. So that's why I kind of think of S and sigmoid. And then we have the end of the GI tract, so the sigmoid colon up here kind of bends, and then it becomes the rectum. So the rectum, the important thing about the anus and the rectum is, again, we have actually two anal sphincters, an internal and external anal sphincter. So one is controlled by smooth muscle, the internal, and then the external is skeletal muscle. So we have control over that. So what are some special features of the large intestine? So if we kind of look, we talked about circular folds and villi in the small intestine. Now these are kind of what the large intestine looks like. So if you notice, there was a band kind of running along the large intestine. So this band of connective tissue is called the tenai coli. And it's just a band of connective tissue, and essentially if you think of it, it kind of puts some tension on the large intestine, and it creates these saccules or sacculations of the colon. And so you'll see what I mean in the pictures when we look at this, and these sacculations are called haustra. And I pointed out the appendix, and even though, you know, it's part of the lymphatic system, we consider the appendix as part of the large intestine as well. So here is our model again. So we have the cecum and the appendix down at the bottom here. And then if you notice, we have this band. So here's a band kind of running along, kind of on either side, but it's more prominent on one side than the other. And so that's the tenai coli. Okay, and then if you notice, see how smooth the small intestine is, you know, it makes its turns, but notice that this large intestine is lumpy, right? So it has these lumps, and we call those sacculations haustra. Okay, so each sacculation is a haustra. Okay, so those are some special features, how you can kind of tell, okay, this is large intestine versus small intestine. So now if we remember way back to our first lecture, we were talking about our serous membranes. And we talked about the serous membrane that is in the abdominal cavity, and that's the peritoneum. So the peritoneum, or the peritoneal cavity, is one of our serous membranes. So it really surrounds and lines the abdominal viscera so that they can move in the abdominal cavity with less friction. So remember, with all of our serous membranes, we had two serous membranes, the parietal layer and the visceral layer. So the parietal layer would be the one on the wall of the abdomen, the abdominal wall, and then the visceral layer is going to be the one right up against the organ. And the The thing about the peritoneum are we create these folds in the peritoneum in the abdominal cavity. And so we'll talk about these folds. But we talked about that they also tie down the organs so that the intestines will kind of stay in place. They can move, but they'll kind of be tethered down in one location. So it reduces friction, but it also holds the organs in place a little bit. So we'll talk about that. So because the peritoneum is so large and we get these double folds in the peritoneum, we have some that we have named. So on the front part of the stomach, the greater curvature, we have a big peritoneal fold called the greater omentum, and it looks kind of like a curtain. So we'll show you some pictures of that. And then in these different regions of the small intestine, we get kind of these double folds back to back, like a peritoneum on either side. So you have 4 layers, right? So 2 visceral layers, 2 parietal layers kind of back to back. So we have the mesentery that holds the small intestine in place and the mesocolon that holds the large intestine in place. So hopefully the pictures will help explain it a little bit more. So if we have a drawing here of a mid-sagittal section through the abdomen, so let's take a little tour through here. So if we find the liver at the top with the diaphragm up here, that's going to be the start of our abdominal cavity. So then we find the stomach here, right in the middle. And essentially we have this flap that comes off the greater curvature of the stomach, and it's going to come all the way down and essentially make a turn. So it folds on itself and then comes all the way back to the dorsal side of the abdomen and tethers us back to the back of the abdomen. So that little curve in the front is called that greater omentum. Okay, so it kind of just covers like a curtain all the abdominal viscera underneath. So then if we look, here's the transverse colon right underneath the stomach. Okay, and it's going to have a layer of peritoneum behind it. And if you notice, see it's got 2 sides, so 2 visceral layers there of the transverse mesocolon. So the mesocolon is the double peritoneum, and it comes up and tethers kind of back where that greater omentum does as well on the dorsal side of the body cavity. And then we have these loops of small intestine. So you see all these circles down here. These are all those loops of small intestine, and they all tether into the mesentery. Okay, so this mesentery comes up, same thing. It kind of comes around the duodenum. We can't forget that part of the small intestine, and ties back up here as well to the dorsal body cavity. So again, it tethers all of these organs in place so that they can kind of move freely in the abdomen without friction, but they're not going to go somewhere they're not supposed to. So here's, it's not the best picture in the world, but you can kind of see the stomach up here, and on that greater curvature you have the greater omentum covering it. Okay, so it kind of looks, it has a lot of fatty tissue in it, so it's kind of yellow and it has this kind of lacy appearance, which is kind of weird looking. So that's the greater omentum. So if we look at this picture of the large intestine, we have all the other intestines removed. We just have the large intestine. You're able to appreciate the mesocolon. So that's the peritoneum coming off the back of the colon. So just you don't need to know the different regions of the mesocolon, so it just is named for the different regions of the large intestine. So that top part is the transverse mesocolon, etc. So, but just know this is all the mesocolon tethering the entire colon down to the back of the abdomen. So on the model, if we look at our torso model, you take the stomach out and all of that. You see the spleen over here and the pancreas. Here is the mesocolon. So it's tethered to the back of that large intestine here. And even though the stomach is removed, this would have been that greater curvature of the stomach right here. So the omentum, the greater omentum, is attached to that and kind of folds over the front of all the other intestines. So we've talked about all the organs that make up the tube of the GI tract, but we also have structures called accessory organs. So accessory organs are really important for digestion. And we call them accessory because they usually don't come in contact with the food, but they secrete some sort of digestive enzyme or juice to help with breaking down the food chemically breaking down the food. So the pancreas, the liver, and the gallbladder are some of our major digestive accessory organs. And we have some in the mouth as well. So teeth and salivary glands and the tongue are all considered accessory organs. The teeth and the tongue kind of don't follow the rule with not touching the food, but they do help to mechanically break down the food. So this is just a picture of those accessory organs. So you take away all the other digestive organs of the tube, and you notice all these accessory organs are all kind of sitting around the stomach. So we've taken the stomach away, so the stomach would have sat kind of right here. Okay, so if we take that away, right, we can see the pancreas sits under the stomach, the spleen and the liver are all around it, the gallbladder, and all of these ducts from these different organs are going to all enter the duodenum. Okay, so we'll talk about that. So first, let's talk about the pancreas. So we've talked about the pancreas before having some endocrine function. So the pancreas produces some hormones that help control our blood sugar levels, but most of the pancreas actually has exocrine function, right? So it's these acinar cells are going to produce and secrete enzymes, and we call them the just pancreatic juices or pancreatic enzymes. And they're going to flow through the pancreatic duct, okay? And then it's going to join in with the bile duct from the gallbladder and the liver, and it's going to dump into the duodenum via the pancreatic ampulla. So hepato for liver, and then pancreatic, and then ampulla just kind of is like a little opening. Okay, so that's how we get all these digestive enzymes into the duodenum. So let's take a look at the model. Okay, so here's our spleen on the right, okay, and then our pancreas in the middle, right under the stomach, and then here's that first part of the small intestine, the duodenum. Okay, so here is that pancreatic duct running right through the middle of the pancreas, and it's going to come over here and open up into the duodenum. And you see the bile duct, and I'm sure I'll have another picture. It kind of comes behind the pancreas and joins up and dumps in at the same spot. Okay. So if we take a closer look at another model, this is a better picture for that hepatopancreatic ampulla or sphincter. The sphincter comes before the ampulla, but don't worry too much about that. Just know that here's the pancreatic duct coming in, and then here's the bile duct coming in as well, and they join here at that hepatopancreatic sphincter or ampulla. Okay. So the sphincter just regulates, when it's going to excrete the fluids into the duodenum, and the ampulla is just the little opening. Okay, and this is also a good picture of those circular folds within the small intestine. So it just increases the surface area. Now, when we talk about the liver, Liver is quite large, so we actually have 4 different lobes, the right lobe and the left lobe being the largest lobes, and then we have 2 smaller ones as well, called the caudate and the quadrate lobes. Okay. And then we do have a ligament called the falciform ligament that kind of extends from the anterior side of the liver to the body wall. And it's a remnant structure of the umbilical cord. So if we take a look at the model of the liver, the right lobe is definitely the largest. So this whole, right side of the liver is all the right lobe. You can see the gallbladder here sticking out underneath. Now we have the smaller left lobe. So this is the anterior side of the liver. And then this line here in between the 2 lobes is the falciform ligament. I think on the underside it shows a white line, which is a little bit more helpful. So if we look underneath, so we've flipped it over and we're looking kind of at the inferior side of the liver. So this is going to be the anterior side. This would be the posterior side. Okay, so right liver lobe, left liver lobe, and then sitting in between kind of the gallbladder and the left lobe is the quadrate lobe. Okay, and then in between the inferior vena cava and the left lobe here is the caudate lobe. So the caudate lobe is caudal. So we talked kind of about rostral and caudal as being some more directional terms, and caudal meaning towards the tail, or kind of, you can think of it more a little bit like posterior as well. So that's the caudate lobe. So these 2 smaller lobes you can only appreciate on the inferior side. And we can also see the falciform ligament a little bit better. So it's this white line structure. Okay? And the gallbladder sitting there as well, kind of sitting embedded kind of near that right lobe. Okay, so now we're going to finish up with the liver, and we're going to look at the vessels coming in and leaving the liver. So the ones that are coming in are going to be bringing in that nice oxygenated blood, the hepatic artery, and we're also going to bring in all that nutrient-rich blood from the intestines via the hepatic portal vein. So these 2 are coming in, right? So they're bringing blood into the liver. Into the cells, into those hepatocytes for processing, nutrient exchange, all that good stuff. And then once that happens, it's going to leave via the central veins. So we'll talk about the central veins. They're the smaller of the veins, draining the different regions of the liver, and then they're all going to join up into the hepatic veins, which then join up into the inferior vena cava, going back to the heart. So let's look at the vessels on our gross liver model first. So we have an area where all the vessels and bile ducts are coming in and going out. And we call that area the porta hepatis. And I know that's not on your list, but essentially it's just this region here with these 3 structures, okay? So we see our hepatic artery coming in, so that's our big vessel bringing in all that oxygenated blood. And then we have our hepatic portal vein, which is why it's purple, so it's a different color because it's deoxygenated blood, but it's that nutrient-rich blood from the intestines. And then our third structure here is the common bile duct. So we'll talk a little bit more about kind of the passageways of bile in a minute. But so we have these 3 structures, and these 3 structures are going to continue throughout the liver. And we call it the portal triad. So we'll talk about that as well when we talk about the microscopic structures of the liver. So once all the nutrient exchange happens, all that good stuff, all the blood is going to exit the liver via the hepatic veins. So they are in blue. So they're the bright blue structures. Okay. So when we talk about the liver, so we talked about blood supply coming in and going out of the liver, but another big function of the liver is to produce bile. So the hepatocytes, the liver cells, are going to be the ones that produce the bile. So a lot of people get this confused because people think the gallbladder is what is responsible for producing bile, but it's actually the liver. And then the bile, once it's produced, if it's not needed at the time, it's going to be stored in the gallbladder. And then once you eat a fatty meal, because bile helps to digest fat, the gallbladder will secrete an extra amount of bile into the small intestine to help digest that fatty meal. So there are some bile ducts. So we have some ducts that are called hepatic ducts because the bile is produced in the liver, and then we have to either get it out of the liver or go to the gallbladder. Okay, so we have the hepatic ducts bringing the bile out of the liver, and then it's either going to go to the gallbladder via the cystic duct or go out of the liver altogether via the common bile duct. Okay, so let's take a look at those different structures. So this is our same picture from before. We have our porta hepatis with our three structures. So we talked about the vascular side, but let's talk about the bile side. So here's our gallbladder in green. So this large structure here is our gallbladder. Okay, so all the cells, the hepatocytes, are producing bile, and then they're going to be drained through either the common hepatic duct, if you're coming from kind of this side of the liver, okay, or you're going to come through these right or left hepatic ducts, so they drain here, okay, and they're all going to merge right at this common bile duct. So they're either going to leave the liver via that common bile duct, or they can go up through the cystic duct and stay or be stored in the gallbladder. And then if we eat a big meal, the gallbladder can then excrete that bile out that cystic duct and out that common bile duct as well. So again, we're going to produce it in the liver, store it in the gallbladder when needed, and it's all going to come out that common bile duct down, join that pancreatic duct through that hepatopancreatic ampulla into the small intestine, the duodenum. So now if we look at the actual, cells of the liver, we look at how the liver looks under the microscope, we see some of these cool structures. So we talked about the cells being called hepatocytes. So those are the cells of the liver. They're the ones producing the bile. They're the ones, cleansing the blood and doing all the stuff with the nutrients. They're going to turn sugar into glycogen for storage, all that good stuff. They're going to be, the hepatocytes have lots of jobs. And the sinusoids are where all that blood is going to be dumped. So the arterial blood and the portal blood is all going to be dumped into these sinusoids, which are going to flood the hepatocytes with all that nutrient and oxygen-rich blood. And so when we look at the liver, it's going to be kind of sectioned off, and that's where this portal triad is going to come into play. So you have the artery and the portal vein kind of bringing in the blood supply, and then you have the bile ductules, which are the small little bile ducts leaving the areas of the liver. So let's take a look at that on the models and pictures. So let's start with a picture first. So if we blow up just a little region of the liver, what happens is the liver creates these lobules. So they kind of look like a hexagon shape, right? So they have a couple of sides, and on each corner of those sides, we're going to have a portal triad. Okay, so on each corner of these little hepatic lobules, you're going to have the portal triad. So you're going to have an artery coming in, you're going to have a portal vein coming in, and you're going to have a bile duct going out. Okay, so that's your portal triad, and I want you to know what makes up a portal triad, okay? So you bring in all that arterial blood. So if we blow up just kind of a corner of one of these lobules, we're going to look at this picture on the right. Okay, so here's our hepatic artery, our portal vein, and our bile duct. So that's our portal triad. Okay, so the artery and vein dump into those sinuses, sinusoids. Okay, you can kind of think of it like a sinus, so sinusoids, which we said sinusoids are leakiest types of capillaries. So the sinusoids are going to be super leaky, let all the nutrient gas exchange happen. And then once that happens in these little hepatocytes, so each of these little tan circles is a hepatocyte or a cell, a liver cell. So once all that exchange happens and all of that blood is now going to be venous blood, and it's going to dump into the central vein. So the central vein lives at the center of all these hepatic lobules, all these hexagon-shaped structures, okay? And they're all going to drain into the hepatic veins and leave the liver and go back to the heart, okay? And at the same time, so you have blood coming in, right? So blood coming in, draining out. You have the production of bile in the hepatocytes being produced, and so they're going to exit the liver via those bile ducts, okay? So again, blood coming in, bile going out. Okay, and then on the left here is an actual picture of what the liver looks like. So we have a portal triad. So we have our little hepatic artery right here. We have our larger in diameter portal vein coming in. You can see all the erythrocytes in that portal vein. And then you have the bile duct, okay? And remember the ducts are lined with that, those cuboidal cells. So that looks like a duct, right? So you have your portal triad, 1, 2, 3. And all these other cells are these hepatocytes and hepatic sinusoids over here as well. So that's hard to identify under the microscope, but it's kind of cool as well. So here's just another picture showing what's happening, and I like this picture. It blows it up really well. So you have that arterial blood coming in, that portal blood coming in, all merging in that sinusoid, lined with all these hepatocytes producing bile as well. So that's all going to come out via the bile duct. So blood coming in, bile going out. And we call these little bile canals canuliculi. So it's just another term for these little tiny bile ducts, okay, draining all these little lobules of the liver. We do have a model in lab as well showing this structure. So on the left is the bigger part. So you can kind of see these, the hexagon shape in cross section, but it's kind of like a tubular structure when you're actually talking about 3D in the liver. So again, you have these hexagon lobules, and at each corner, you're going to have a triad. Okay, and then in the middle, you're going to have a central vein. Okay, and so it blows up that corner as well. So you have your triad, then you have your sinusoids running in amongst those hepatocytes, and then you have the little canuliculi or little bile ductules coming out through that bile duct. That's kind of cool. So that's it for the digestive system in lab, and we'll talk a lot more about it in lecture as well. All right, adios.